Hi, everyone. Well, thank you for joining our webinar today titled, How Does Particle Characterization Enhance Research and Scale Up, Validate Production, and Increase Your Bottom Line? With me is your speaker, Dr. Willie Hendrickson, the founder and CEO of AVEKA Group, one of the most popular speakers here at our HORIBA webinar program. So if you haven't listened to the 3 other webinars that Willie gave in 2022, ping me and I'll be happy to send the recording to you. My name is Julie Chen Nguyen. I am your facilitator and I'll be monitoring any questions and comments that you have in the background. And then we'll have a Q&A session to go over those at the end of Willie's talk. I think now is the time that I pass the ball to you, Willie. If you're ready, please go ahead and share your screen.
Thanks, Julie. Thanks for the organization, and thank all of you for joining us here today. As Julie says, I'm going to talk about particle characterization and how it can help all of us in research and production and bottom line. So let's get started and see what we can do here. So I'm going to give you just a very short overview of AVEKA. This is the company that I work for. You know, I have a general question. Why do we care about particle processing? Some of the general challenges for industrial particle processing. I've got one example of economics of particle processing, which is really going to lead us right into characterization. Lots of examples, future directions, and conclusions. So let's get going. The AVEKA Group, we are a contract manufacturing company. We are focused on particle technology. So, you know, giving these talks with HORIBA is always an exciting time for me, because we're really talking to you, the people that are knee deep in this area. AVEKA is a spinoff from 3M from 1994. We have 5 manufacturing plants and currently have about 280 employees. We actually look at ourselves as a different type of contract manufacturing organization. We, as opposed to many contract manufacturers, we have a broad range of unit operations in industrial areas. We have a very active research program, and we're really into technical and collaborative business model. You know, to just give you an idea what we see every year, we get about a little over 2,000 inquiries a year. It's a lot. We get inquiries from the food area, from the battery area, from areas that, you know, don't even understand them half the time. But, you know, out of this, there's 600 project proposals that we write a year, and we're dealing with 300 customers a year in, you know, areas that span just about every industry area. Now, you know, the question is, why do we even care about particle processing? You know, at the end of the day, there's been some people at 3M, at DuPont, at Dow, have looked at their products, and they've actually stated that, you know, somewhere between 60 and 70% of all products have either a particulate nature or are made with particles somewhere along the way. And, you know, and that's really true. I mean, you know, for those of us that are in particles, we really understand this. But let me just give you a couple examples of really big impacts in particle processing. I've listed 4 of them here, but I'm going to talk about 2 of them today. One of them is in the 1930s, Minnesota, where I'm from. We've been the center for iron mining in the United States for a century almost, and we found out that the high-grade ore was running out. Somebody looked around and says, well, you know, we got a lot more low-grade ore, and so at the University of Minnesota, they put together a program to grind up this material, this taconite ore, beneficiate it, take out the iron ore, and then you know, take those 10 to 20 micron particles and send them off to a blast furnace. Well, what they found out instantaneously, 10 to 20 micron particles in a blast furnace shut down the blast furnace. You couldn't push the CO through the system, so you could get no reduction, and you couldn't, you know, produce iron metal out of this. So they went back, thought about it a little bit, they agglomerated the particles, they sent it back to the blast furnace. Lo and behold, something else happened. Instead of it shutting down the blast furnace now, with these uniform particles that they've never had with standard iron ore, increased the throughput of the blast furnaces by a factor of 5. You know, when you consider the amount of energy it takes to make iron metal from iron ore, you know, in today's world, this would be looked at one of those great green energy projects that you cut out 80% of the energy to make iron ore. You know, and people just thought, well, you know, this is a standard process, you know, this is how we improve things. But the reality, that's such a big step, and it affects everything in our society, that we can have better, cheaper, faster iron to build whatever we need to build. Fast forward to these past couple of years where we've been in this pandemic, particle technology with the development of, you know, really specific liposomes for that mRNA delivery, you know, played another big part in a significant change in our society. So, you know, I could talk about xerography and cement manufacturing and energy costs, but, you know, I think you get the picture that how important, just in general, particle technology has been and continues to be in the world. For industry, we've got some big challenges in industrial processing, and I've divided them up into two areas here. One is operations. You know, in operations, I really think there's economics and technology readiness levels. I'm going to talk about economics in a couple slides from here, just to give you an idea where this really affects us. But from a technology standpoint, I mean, there's lots of technology areas that are a challenge, but I've picked three: water, powder flow, and grinding efficiency. And so, you know, when I talk about water, I like to say I hate water, because I'm either drying it and it costs a lot of money to dry it, or if you look up at the one slide here that shows the agglomerated powder, you know, it causes caking. You know, water is just a problem. Powder flow, all of you might have seen hammer rash before. You know, there's some spot in your plant that the operators will beat the heck out of the pipes to get the powder not to stick there. You know, this powder flow issue, you know, many of the times that we got everything working right, but we can't get the powder from one part of the plant to another. We can't get out of a bin. Huge challenge. And grinding efficiency is also a real challenge. These areas, I'm going to touch on some of these as we go through here today, but just want to, let's set the stage. You know, why do we do particle characterization? We do it to look at these technological problems and how we can make our operations better. Well, let's just take one example and look at economics. I'm going to give you three examples today of spray drying. So this is a good one just to look at from an economic standpoint. For those of you that don't know, this is a cross section here, the little schematic of spray drying. Basically, spray drying is we're taking a solution, atomizing it into a chamber. It atomizes. It's a hot chamber. It dries, and then it's collected either in a cyclone or a bag house, a filter of some sort. This structure, this schematic that I'm showing here, this is very similar to the schematic that you'd have for a Buki spray dryer, one that you have on your lab bench. And you know, you can do some great things in a little spray dryer like that. But the reality, the picture at the bottom of this slide, this is a picture of one of our bigger dryers. These things are a little bit bigger than a lab bench dryer. They're typically 80 feet tall. They cost $30 million to build. It's usually the installation cost that's more expensive than the piece of equipment. And, you know, we're looking on these big dryers to get, you know, over $500 an hour. And we're looking at good yields, but, you know, the parameters that are important to us are, you know, what the input solids are that we're drying and the drying rate. So let's just what considers what happens from an economic standpoint if you change the percent solids in your dryer. If you operate at 1% on a 2,000 kilogram per hour dryer, you're going to be able to dry 20 kilograms an hour. Now, if I'm charging $500 an hour from my dryer, I'm going to charge this person just for processing $25 a kilogram. However, if I operate at 50% solids, I'm going to get out 2,000 kilograms an hour. And if I want to make my $500 an hour, I'm going to charge 25 cents a kilogram. I mean, this is pretty important. You can see this big change here from 1% to 50%, but let's consider a real application that we're all kind of faced with. We do a lot of drying in my company at 20% solids. So let's assume that we're operating with our 2,000 kilogram per water hour water evaporator. We're operating at 20% solids. We can get 500 pounds of product, dried product out of this dryer theoretically. But you know, if the percent solids fluctuates by a percent, just 1%, it changes, you might drop off 31 kilograms an hour or gain 32 kilograms an hour. Now, in one sense, 32 versus 500, that doesn't seem so bad, but a typical dryer will operate for 7,000 hours a year. I mean, that's a low operation. A lot of them operate 95% of the year at 8,300 hours a year, but just at 7,000 hours a year, the difference in pounds of product that we can make is 3.3 million to 3.7 million. So by having no extra costs in manpower, no extra costs in energy, you can get another 400,000 kilograms of product out. And at $1.50 a kilogram, that might be $600,000 that can go right to your bottom line. This now becomes real money. So just these very, very small changes can have a big effect. The point of me talking about this, and for all of this prelim, is to really say the way that we try to control how we're maximizing our throughput, maximizing the specific aspects of the product that we're getting, is by characterization. So let's just consider what we do at AVEKA. We consider ourselves not only a different type of CMO, but we also consider ourselves a characterization-focused CMO. We have a broad range of characterization methods, and we use it for process control, quality, materials, understanding and development. And we do over 50,000 analyses per year. This is a lot of analyses. Of those 50,000, over 20 of them are particle size distributions, and we're using over 30 different analytical techniques. We focus on this as a way to control the quality, the process control, the optimization. And if we didn't do this, I don't think that we would really understand what we're doing, and we wouldn't be able to gain the advantage that we're really looking, that economic advantage. Here on this next slide, this is just an examples of some of the equipment that we have. We have, we're particle analyzer, we're a particle company. Of course, we're going to focus on particle size analysis, and you can see down here in the lower right hand part of the screen, example of 2 HORIBA instruments that we have. Quite frankly, I'm going to give HORIBA a plug here. These are absolutely incredible pieces of equipment. You should own 6 like I do. But we also do a lot of other things. We do scanning electron microscopes, and it's a picture shown here in the middle. Surface area density, formulation analysis, TGA, DSC, flow characteristics. I think the point is, at least for us, is that characterization is such an integral part of how we manage our business that we can never have enough equipment, and you can never do enough particle size analyses. So with all of that, let's get to the examples. What I like to do in a talk like this is I'd like to give you kind of the framework of where we're going, which hopefully I've just done. But I'd like to give you some examples of how we use particle characterization to help us with all those aspects, from optimization to development to whatever. I've got 4 areas I'm going to talk about today: spray drying optimization, classification efficiency, grinding, and something beyond that I'm calling beyond particle size analysis, particle size distribution. We're going to be looking at density, shapes, yields, controlling the D50, or actually coming up with new methods. So let's get started with this, and let's give you our first example in spray drying. We had the challenge that we had in this particular case is we dry a lot of sodium acetate. This is an interesting food additive that we dry. And because we dry so much of it, sometimes we're drying on one dryer and then we dry on a different dryer. Well, spray drying is spray drying, right? It should be the same. What we found out that it wasn't the same. And in fact, I think all of us know that are in the business of particle processing. Change the equipment, change your conditions, even small amounts, and things change dramatically. So what happened is we were drying sodium acetate on one dryer. We went to another dryer, same material, same supplier, and what we found is the product on one dryer, on dryer number 2, didn't fit in the bag, but on dryer number 1, it did fit in the bag. So the analyst that got this problem, he was asked by the plants, what's going on here? What's different? So the first thing he did is he measured tap density, and not surprisingly, on the dryer number 1, it's a higher density than dryer number 2. Dryer number 2, remember, is the dryer that doesn't fit in the bag. And so right away you can see, yeah, it's about, there's a difference here. Is it enough? The other thing that he noticed is that when he did the tap density at time 0, just put it into the cylinder to measure it, and then tapped it for either 30 seconds or 60 seconds, the density increased, completely expected. Everybody, you tap something that the particles pack a little bit better, but it took longer for the tap density on dryer number 2 to get to the final density, and the density wasn't as great. Now, as it turns out, we know that the tap density of around 0.4, the one that we see at the beginning of dryer 1 and the one that we see at the end of the tap density will fit in the bag. But, it just takes longer to get there. So the analyst did a particle size analysis, and he was actually looking at shape analysis. So he used a piece of equipment called a cam sizer. It actually does a measurement of size and shape at the same time. And what he found is dryer number 1 had smaller particles than dryer number 2, but dryer number 1 had a more spherical powder. In this case, sphericity, the closer it is to 1, the more spherical the powder is. And the sphericity, was that enough? His conclusion was that sphericity was the major contributing factor. Well, let's just see. So I always like, as we've done this analysis here, we can see a difference, but what's causing it and how do we control it? So if you look at the pictures here, this is a SEM picture of the product out of the 2 dryers. And if you look at dryer number 1, you can see spherical particles, a lot of spherical particles, exactly what we expect. And dryer number 2, in concurrence with the sphericity measure, there's spherical particles in here, but there's a lot of really odd shaped, odd looking particles. If we take a look at this at a little higher magnification, you can see in dryer number 1, they're relatively spherical, but you can see some odd kind of structures in here. But if you look at dryer number 2, holy mackerel, what the heck is that, Batman? I look at that and I kind of wonder how, where did this particle come from? At the end of the day, I'm going to come back to this, but I want to leave you with the first thought on this was it was a spherical issue, but we didn't have an idea to begin with why we were seeing the differences between dryer 2 and dryer 1. Very, very odd. Let's go on to the second example, and I will come back to this, and we'll talk about what this really means. The second example, we had a customer that wanted a powder dried to make a spherical powder, and if you can see down here, this is the SEM image of the particle. Now, I'm not showing a particle size distribution of this powder, but we hit the size that the customer wanted. But with great imagination, I could call this spherical, but I really don't look at this as a spherical powder. So what the person who was doing this, the engineer that was doing this, thought, something, what's the size of my starting material? This was an inorganic slurry that was being dried, and when he looked at the particle size distribution, he'd get a particle size of about 10 to 12 microns. A mean particle of 11.7 microns. So he thought, perhaps this particle is too big. What would happen if I ground it? He ground it with a bead mill, and he got a particle size distribution of 1.9 microns. Now, if you look at this particle size distribution down here on the bottom, what you're really seeing is some material that didn't grind, that was still up at that 10 to 12 micron range, and most of the particles went down into the 0.2, 0.3 micron range. That 1.9 is probably misleading in a sense, because from a distribution, a number, you've got a lot more of these 1.9 particles. When he took this material and dried it, he ended up with, going from this kind of granular looking particle to the spherical particles that he was looking for. So by, understanding that a particle size was out of range of what was optimum for this type of spray drying, and getting it into the optimum range, he could take that starting material, make the right type of slurry and dry it quite nicely. Well, let's take a look at our last particle size, or our last spray drying example here. In this case, customer is asking to get a tap density of the final product, a higher density than what they were currently getting, without appreciably changing the particle size. Now, the engineer in this case, kind of approached this in, what can I change? Can I change the feed rate? Can I change the percent solids? Can I change the temperatures? Can I change the atomization technique? And he looked at quite a few of them. At the end of the day, most of the changes really didn't give the effect that he wanted, but changing the percent solids did. So you can see here, if at the top particle size distribution, he spray dried at 23% solids. He ended up with an average particle size of 11.4 microns. He's got, a large and a small peak here. And the tap density was 0.28 grams per cc. If he increased it up to 30% solids, the particle size, distribution, the mean change, it went up to 19. That was still considered reasonable within the scope of this work. And his density went up to 0.37. Now you got to ask yourself, why did this work? Same material, same drying technique, and by putting higher percent solids, he got a higher density. This is a tap density now, not a true density, but a tap density. At the end of the day, if you look at this particle size distribution down here on the bottom, particularly the large peak, you can see that it's a much broader peak. It goes from, basically 1 micron to 100 microns, where this one goes from 1 micron up to about 60 microns. This difference in size, I believe, allows the particles that are formed to pack better. And so when they do the tap density, you're going to get a higher density. So, interesting, you think about these three examples, three different cases. We're spray drying, but we're looking for three different things. And, you got to ask yourself, are the conclusions right? I mean, we've got characterization data. We have something here, but I'm gonna opine here that there's a little bit more to get, because if you don't know your process along with your analytical techniques, you can be fooled sometimes. So take a look at this picture here. I picked this picture out on the left side of the screen here from an article by a man named D'Souza Lima, printed in Particle Technology, and he talks about what happens when you start out with a drop of liquid in a spray dryer. What type of structures can you get from the final spray dried product? Well, up here at the corner, my vision of spray drying is I take this droplet and I get this nice dense sphere. So on a real, first look at spray drying, this is what you get, and this is the vision that I have. However, we do know that if you dry with slightly different conditions, as shown up here in the upper right-hand corner of this part of the slide, you can get hollow bubbles, and that's shown in this picture here. If the bubbles aren't very strong, they can collapse into wrinkled particles, or they could collapse into donut-like particles. These are structures that you see in spray drying, very common, and it's a bubble that's collapsed. You can get where you have solids in your system. You can get what he calls a raspberry-like particle. Quite frankly, I've never seen a hairy particle. I'd love to see one someday. Or you can get some highly porous particles. So think about the analysis. The analysis that was done by the engineers and the analysts for these three examples that I showed you are absolutely dead on. These are just some examples of particles that we made to kind of show that, yeah, we can make these types of interesting materials. But what I found very, very interesting about going back to this first example is, quite frankly, I have never seen images like this in my life. The size distribution isn't going to tell you what's going on here. The sphericity might tell you something that's going on, but when you look at these pictures, it's like, what the heck is going on here? So let's go back to this slide here, and let me make some guesses what's going on. We actually spray dried a nice particle, and it was probably a particle that was a fairly dense particle like this. But interestingly, sodium acetate has some really interesting effects afterwards. It's hygroscopic as heck. So here's an example of a different hygroscopic particle. This is a spray dried aluminum sulfate that we did. You can see the big particle down here. It's a hollow particle. You can see the crystalline structure inside. And now if you look up at the kitty corner picture up at the top, you've exposed this very nicely crystalline particle, and it's absorbed water, and it's really changed the structure. And in fact, the big particle in the middle, that is this broken particle that's kind of mushed out a little bit, is interesting. But what's really interesting is the particle up in the corner here, where these particles actually agglomerated. They got wet enough in the process chamber or after the process chamber to change shape. So that non-spherical particle that we saw in sodium acetate, it might have been, the effect might have been, the sphericity wasn't right or the size was bigger, but that actually happened by a secondary step that we had not ever seen before. And in fact, when you consider a lot of these analytical techniques that you do, you can do some great characterization and analysis. But if you don't fully understand the process and what the different parameters can be, or how the material can change afterwards, you can be misled. And so, the point of this is lots of techniques can help you, discern what's going on, but you have to really couple that with an understanding of what your process and materials are. So let's go on to classification optimization. In this particular case, I love this example, because this is the classic academic example of working with glass beads or glass bellatini. The academics really like to look at the optimized idealistic systems, and every once in a while we get an industrial problem that actually fits that optimized idealized system. So in this case, we had a collection of glass beads that were made. We were going to air classify them, and we were looking at getting the highest yield that we could get while maintaining the cut points that we were looking at. So here on this slide here, you can see the glass starting bead material. This is a distribution of that starting bead material. And if you look down here, it's got a D50 of about 33, a D10 of 19, and a D90 of 48. But the customer was interested in cutting off that tail and tightening it up a little bit, and they had a vision that they really wanted the cut point to be between 25 microns, a D10 of 25 microns, and a D90 of about 48 microns. And so we did that. We actually did that classification, and I believe that's the darker classification up here on this other distribution. But we looked at this and we thought, what would happen if we just extended the classification just 1 micron? On the low end and a couple of microns on the high end, really not a big difference. But, at the end of the day, it had an incredible effect on the distribution, as shown here on this next slide. So up here in the top left corner, you can see the starting beads, the picture of them. In the top right corner, you're seeing the narrow cut distribution, the 25 to 48 at 43% yield. And when you broaden it out, and it's kind of it's tongue in cheek, I'm saying broaden it out to the broad cut, by only 3 microns, the yield goes up to 73%. Having the right classification methods, having the right characterization methods to actually be able to show this, and show it from an SEM and from a distribution, it's absolutely critical. And the difference between a 43% yield and a 73% yield, that's huge. That's massive. And I think we all know that. But how would you have gotten there if you hadn't really done that good characterization and do it in a couple ways to actually prove that you haven't really changed that much? So let's give you another example. We had a customer that asked us to classify silica. They wanted to get rid of the fines. And so the raw material you can see here has a distribution, a mean at about 10.97. And what we were really asked to was to only take the fines out, only take the fines out. And so what we did is we classified and changed the distribution by cutting off at the low end here. This is an air classification process again. We cut off the low end here, and we raised the particle size up to 11.43. Hardly changed it. And the shape of the distribution doesn't look that different. But it wasn't quite right. So we tried it again. And this was really remarkable. We did a second cut classification, cut the tip off of the low end of the distribution here again. And we got an average size distribution of 12.8. We basically changed the particle size from 11 to 12 microns in a two-step process. If you only looked at the size distribution on here, which is absolutely critical that you know what that size distribution is, but if you only looked at it, you wouldn't see what we saw when we looked at an SEM analysis. So here, if you look at the raw material, you can see the large chunks and the small particles. Here, if you look at the first pass classification, it still has the small particles and the large particles, but look at after that second classification. Basically you've gotten rid of all those fines. And the question is why did we need to do this twice? If we hadn't looked at this from a particle size distribution, we would have never seen it. I think our customer might have been able to tell. Functionally, they were looking for a few fines, and the first cut classification had them. But from an analytical standpoint, seeing a picture sometimes is worth 1,000 words. And in this particular case, you can see we weren't quite there, but boy, after that second classification, truly it got to that clean product that we were looking for. So let's consider a jet milling and classification. So basically, this is an example of a jet mill. The picture here down at the bottom. So we jet mill this material, and then we take the same material and we go through the classifier. In both cases, we're collecting in a cyclone because of the size. And in this case, we're going to take a polymer particle, grind it up, classify out the fines. This was to enhance the flow of this, and you couldn't do this in a one-step process. You had to do both steps. So let's just take a look to see what happens here. So from the polymer, this is the polymer after we've jet milled it. And typically in characterization, you're always looking at a volume distribution. How big each bin is, the volume of this. So, what you see here is we have this long tail of fines, and our customer had already determined that this long tail of fines wasn't good. So what we did is we classified that material and we got rid of the fines. You can see this over here in the upper right-hand corner. Volume distribution, but we did many different test runs on this thing, and we couldn't tell the difference between one cut point and another cut point. They looked about like this, but we couldn't really see. What we did is, instead of looking at the volume distribution, we looked at the number distribution. And we all know if you have a particle at 10 microns and one at 100 microns, the same volume for one 100 micron will take 1,000 10 micron particles to get. And so this is exactly what we see. Could be the same volume here for a 100 micron particle and a 10 micron particle, but the number of particles is incredibly higher. Now, if we take this jet mill material and look at the number distribution after our jet milling and classification, we can see that so long as we kept our parameters below 1% in the number distribution in this area from 8 to 20 microns, the particle passed what was needed for the best flow characteristics. So this is a case where characterization tells you exactly what you want, but you actually have to look at the right type of characterization, not volume distribution, but number distribution. And here's some pictures of the starting material. This is a solution-produced polymer. It's kind of a potato knobby-like structure. This is after the jet milling up at the upper right-hand corner here. And then finally, the jet milled and classified particles here, show the loss of the fines and actually the good functional characteristic and particle flow that you're looking for. Once again, classification or characterization really helps us understand what's going on here, but understanding it in a volume versus a number versus volume. So one of the other challenges that we have is how to grind up materials and do it in the most efficient way. So we've had a process for a number of years to take a ceramic substrate and go through a grinding process to get a tight distribution. And so these ceramic substrates can be large, they can be small, but we've got to grind them down to the same size, and we have to do it consistently. So what we were looking to do is see if we can reduce the grinding time and also tighten the distribution from a starting D50 of 15 to 35 to 17 to 19. But what we did to do this is we went through a 3-step grinding process, and this is not unusual. If you're going to start out with a grinding process and you want to get everything the same, kind of take it down from one size in a crusher, take it down to another size in a hammer mill as shown here, and finally use a ball mill and grind it up to get your final size. And this is exactly what we did. This is an actual picture of the process that we use. This is the crusher. This is an actual cone type of crusher, followed by a magnetic separator, followed by this blue device here. On the left side is the hammer mill that we have. We'd screen this material and then we'd send it over to a ball mill. So a crushing, a hammer milling, and a ball milling process. As it turned out, this takes a long time to do. You just can't be changing your process, in a random process. So in 2003, when we started out this project, we were making product that had a 13 to 35 micron. This was a lot of different sizes, comes from a lot of different places. We thought that we were doing pretty well in this 13 to 35 D50 by crushing and ball milling. But by 2006, our customer was looking for, can you do better with that? Can you get it down to 18 to 24 microns? Turned out that a hammer mill and a ball mill was the right way to do it. So, we put that in. By 2013, they were looking for even a tighter distribution, 16 to 19 microns, and this came into the crushing, hammer milling, and ball milling stuff. But the interesting thing is in this time as we were doing this, we were literally doing thousands and thousands of particle size measurements because we were measuring off the crusher, off the hammer mill, off every ball mill run that we were doing. And what we noticed is that there was a seasonal cyclability on the particle size distribution. We were within the specs that we wanted to, but we wouldn't be able to improve that spec unless we understood what was going on. So with thousands of measurements and plotting this all out, we've determined, and not surprisingly, this is my old friend water, is that as the relative humidity in the room changed, the particle size distribution changed. And so what we did is we added a grinding aid. We added a little bit of water to each one of our ball mills as we were doing this. And all of a sudden, the grinding rate got better and the distribution got tighter. Now, what we think is going on with the grinding aid is the grinding aid enhances the fracturability and actually shortens the grinding time. So this is a great energy conservation, but it grinds it faster so you don't overgrind and have your distribution doesn't spread out. We thought we had this thing absolutely positively figured out. But as you can see at the bottom, there's a caveat here. And the caveat is that with processing, with characterization, it's never done. So as we look at this next slide, what we found is that when we ground material, and I have two distributions, particle size distributions up at the top, for all intents and purposes, as far as I can tell, they're exactly the same. But in one case on the left, that sample passed the final requirement, but the one on the right didn't. And what we found out was that, and this is not surprising, particle size distributions don't necessarily show you if you have a very, very small fraction of a particular size, of one particular size. And so what was happening is we did all these grinding steps. There was always a few particles, and I truly mean a few particles in every 300-pound batch that were bad, that were too big, that were outside the range that we were allowed. We couldn't see them with the standard laser diffraction system. So what we had to do was drop back and wet screen about a pound of sample out of every batch that we did, and literally count up the particles on that screen for us to see what was going on. So we had great characterization, we had great understanding, we improved the process by having all this characterization, but at the end of the day, it still came down to a visual measurement. So, you know, all the great characterization methods in the world don't work if you don't look at your particles. Most of the time we like to look at them under a microscope, but sometimes you got to count even the smallest ones that you, smallest amount that you possibly have, because that could be the kicker. All right, let's change to characterization beyond particle size. We see this all the time, and you know, we love particle size measurements. I mean, it's the way that we really think as particle processing people. But every once in a while, you get something that's outside that range. You need more than that. So we had a customer come to us and ask us, can you produce a cellulose fiber with a high water holding ability? They had the process all figured out. We actually replicated their process. We scaled it up, replicated it. And basically what we were doing is we were removing lignin and hemicellulose from corn bran. So corn bran's the outside shell of the corn that's left after milling processes. The process was fairly simple in description, but very difficult to do. You wash in caustic and you centrifuge, and then you finally dry. And what happened with this process is we found out fairly quickly the process the customer brought to us wasn't optimized. Surprise, surprise. And actually, even worse, the product did not produce purified cellulose like they want, but microfibrillated cellulose. And so what happens when you look at cellulose? Cellulose is a long particle with a hollow lumen. That's where the water flows when the plant is growing. But on the outside, the cellulose structure is a triple layer of cellulose microfibrils of cellulose, shown here in the SCM. And when you beat on this material, you can actually spread this out and you can fibrillate this stuff and get this material that is very high water holding. So we didn't know that, though, to start with. We had yields that weren't right. We had incredibly inconsistent process. The water holding results were inconsistent. You know, what did we do in a case like this? Now, we could have gone back. We did go back and blame the customer. The customer blamed us. And at the end of the day, we decided, you know, we're going to need to analyze this and understand this. You know, what is going on with this process? So what we did is the analysis of cellulosic materials is very difficult. There's wet processes to do these. It takes about a week to do one experiment, one analysis. We needed to come up with a better way of doing it. And what we came up with is we looked in the literature and found out that TGA had been used to analyze lignocellulosic materials, and it was reported that they had these big broad peaks that showed decomposition of the cellulose and the hemicellulose, and they were different from each other. Well, the interesting thing was these papers that we found were published in the 70s, and lo and behold, in the 50 years that had happened since those papers were written, and when we were doing the work, the pieces of analytical equipment had gotten substantially better. So they actually could decompose at a much more controlled rate. So instead of getting a big blob of a peak when you do a derivative of the weight loss, we could actually get incredibly sharp peaks that look like NMR or IR. So we found out that cellulose decomposed, just tightly decomposed at 300 degrees with the TGA, and hemicellulose decomposed at around 270. So now we could tell the difference between cellulose and hemicellulose and actually talk to our customer about what's going on with the processes that we were doing. And so when we took corn bran, that's a mixture of hemicellulose, lignin, and cellulose, we could see two peaks at around 252 to 260. One peak at 325, the 262, 252 was hemicellulose, the 325 was cellulose. And when we cleaned it up and we did the process, the caustic wash, we still had hemicellulose. What the customer didn't realize, because he had never done a characterization, he wasn't making purified cellulose. He was changing the structure of a hemicellulose cellulose structure and getting this new peak that had the high water holding capacity if he microfibrillated it. This allowed us, by doing this characterization, developing this characterization method, to change from a yield of 20% to a yield of 80%. It was incredible, just by doing characterization and understanding. I have one last example for you. One of the things that we get challenged with is doing lab experiments and then scaling up. And this is always a challenge. Do you know that your scaling up process is going to work? In this case, what we were doing is we were taking a number of organic materials, we were heating them, melting them, and we were going to prill them. And prilling is a melt atomization process that we do. But we needed to take 3 materials, blend them together, and prill them all at the same time. One of the things that we wanted to know was, if we scaled this up, how big a heater would we need for this scaling up process? So the engineer in this particular case took a DSC measurement, differential scanning calorimetry, and measured the amount of heat that it takes to melt each one of these materials. And this is shown on this next slide here. So what he did is he measured the component 1, 2, and 3. Each of them had a different DSC curve. Each of them had a different amount of heat that was needed. And what he did is then he also measured if he took all 3 of them together and alloyed them together. Now, his thought on this process, that if I just take the DSC of these 3 components, add them up together, I should get what I need. Well, let's see what actually happens when he did that. So here's the alloy DSC. This is the blue part is the 3 component added together. And you can see it would tell you a relative amount of joules per gram that you would need to do it. But whereas the mixture itself was much lower. So, he could have guessed at how to scale this up. He could have guessed that, let's just take the energies for the 3 and add them together. But this was very interesting. He found out that he needed less heat for the alloy than he would have needed for the 3 ones he predicted. The interesting part about this is characterization wasn't used to find out what the size was, but it was used to actually help you calculate how to come up with the best optimized process in your scale-up process. I love this. I love this example because it helps us get to manufacturing much quicker. So let's think about what we talked about. For industry, it's always the economics. How can we get that extra 1%? How can we change the distribution of what we're doing? Can we expand it? Can we contract it? How does it affect the yields, the rates, the whatever? How can you optimize your new process, your new materials, unless you do extensive characterization? Really should make you really think about this. From a characterization standpoint, I would never use one technique. I highly suggest that particle size distribution along with some microscopy technique, or these particle size distributions with the tap density, or et cetera, et cetera, et cetera. It gives you a much better understanding of what's going on. The images are always interesting, but don't be misled by them. And remember what Ed Merrow says in the report that he did in 1988 when he estimated startup times for solids processing plants. Solids are so much more difficult to do that all the characterization that we can do can only help us be better at taking care of our management on how we're providing the data that everybody needs to make a good economic decision. So where do we go from here? Julie put these webinars that we're talking about. Please think about this. Send us some thoughts of something that you'd like to hear. This industrial challenges of particle processing. Thank you all for voting for that. It's a very interesting talk, and I think you'll enjoy it. But if you have any other topics that you'd like to hear, I'd love to hear them. The other thing I'm gonna put a plug in here for the International Fine Particle Research Institute. I am the president of that organization. It's a consortium of industrial companies that are really putting their heads together, trying to figure out best practices and having good talks and networking. We fund research projects that, you know, many of the areas that we talked about here, we're in this talk. You know, I'll be sending you some information about IFRI, but it's something for you really to consider if you're going to be the best that you can possibly be in particle processing. So the summary, I love this area. I think we're in the most exciting field in the world. I mean, can we go from, where else can somebody go from iron ore to COVID-19 viruses with xerography in the middle? This is an incredibly challenging field. I think all of us, as we do this, we find out that you know, how difficult it can be. But you know, run as many characterizations as you can. It's going to help your profitability. It's going to help your understanding. And let us know if we can help. You know, we love this area. You know, I appreciate you listening to the talks, and I'd be happy to take any questions that you have. Thank you very much.
And thank you, Willie. That was a really good talk. You always get to the bottom line, which I really enjoy, down to the dollar amount per hour. Okay,
Try.
so and I definitely agree. Particle is very interesting. You go from chocolate to virus to battery materials. So let's get to the questions because quite a bit of those came in during your talk. Okay. All right. So question one, kind of going back to your previous slides, what tap density instrument did you use?
Oh, that's a great question, and I don't know. If the person has that, I'd be happy to send that information to them. I just don't have it off the top of my head.
No worries. So I think this is a good time for me to kind of send this general webinar info address to everyone, so you can contact us or kind of follow up any questions that you may have by sending us a quick email to labinfo@horiba.com. Okay. And then question two, what other techniques have you used for fine characterization other than laser diffraction and SEM?
Well, we also use quasi-elastic light scattering, which is, you know, a type of laser diffraction. That's a very good one. But those are probably the three. We've used, you know, the cam sizer, which is a slightly different technique than Fraunhofer diffraction. And we've also used a Coulter counter, which is an L-zone type of technique.
Thank you. And that kind of leads me to the question that came in during registration, which is, what are different techniques and instrumentations that AVEKA carries?
Well, I had a list of that, but you know, Julie, you know, we have lots of HORIBA instruments. You know, we love that, but we also have the Coulter counters. We also have the cam sizers. You know, the SEM, the digital optical microscopes, the QA DSCs and TGAs. We do surface area measurements, lots of moisture balances, OHAUS and Keldal water measurements. I'm sure I'm missing some. I'm missing all my rheological equipment that we have. Zeta potential. You can't have enough analytical equipment. I can't impress that on anybody. I should be buying more every day.
And I would imagine that all these techniques are also listed on AVEKA's website. Is that correct?
Yeah. Yep.
So for sure, you can visit AVEKA, aveka.com for a list of comprehensive technologies that they carry and service that they offer. Okay, the next question is, what are the benefits of implementing particle size analysis in the sugar refining industry?
Wow, in the sugar refining industry, I mean, since that's a crystallization technique, I would think that if you wanted to optimize that, I would sure like to know what your crystallization agent size is. I mean, was it the calcium oxide that they put in to crystallize sugar? Knowing that size, I think could be very important. And I think knowing the size distribution of the crystals as they crystallize could be incredibly important, knowing what's going on. So I would be measuring particle size measurements all over the place in that plant.
Okay, the next question is, how is the number distribution calculated? I think this person's referring to the slide where you converted volume-based distribution to number-based distribution.
That's a great question. I don't know. Julie could probably answer that question better than Willie could answer it.
Yes, and it's a very loaded question. So I'll probably follow up um, with an email afterwards. But basically, we recommend when it comes to number to volume conversions, there's a way that we prefer to do that that's a little bit more accurate. Okay, the next question is, how do you take care of all the extra fine generated during ball milling?
Wow, that is a great question. Ball milling has a tendency to give a longer tail. I actually have an example that I've used other times that show the difference between jet milling and ball milling. What people do where fines become very important in ball milling is they'll actually go from uh, a batch process to a swept ball mill, so it's a continuous process. They'll move the finer particles out of there so they don't become overground. So the way that you deal with it is you minimize your grind times, either by removing the material very quickly or by just doing a short grinding time.
Thank you very much. Okay, the next question, what is wet screen? We have a similar issue where some large particles in the sample that don't show up in laser diffraction.
So what we do in a case like this is you take a volume of material. For us, it was 1 pound of material. We now take that material and we slurry it up in water, and you can screen at a very fine size. So you'll pick a smaller size than you would normally do dry screening. And now you just screen and you're trying to get all the particles to go through. And at the end, you can actually count the particles that stay on your screen. That's the way that you do it. Make a slurry, screen it, keep adding water till everything's washed out, other than the big particles, and then you can count them.
Thank you. What about in the pharmaceutical industry? So I would ask that can you ask what specific questions that you have about the pharmaceutical industry? Is it how to enhance, scale up in the pharma industry? And then back to the wet sieving. What if you don't want to use water?
Obviously, you could use a solvent. It's just the issue of flammability issues then, and are you going to recover the solvent? Is it appropriate to recover the solvent? So any liquid will work. Just be careful about the flammability issues of having a lot of solvent around.
Thank you. So this person actually said the product has to be dry. So I would love to work with you on your application, maybe offline, to maximize so you see large particles using dry jet milling, jet powder dispersion accessory.
I'd love to talk to that person one-on-one. So if they can reach out, I'd be happy to talk to them.
Okay. Any idea on how you can measure particle size on brown sugar, which contains about 3% moisture where particles are sticking together?
The way we would handle that one is we would probably use a HORIBA system, and we would do it in hexane.
I agree.
Yeah, that would be the best way to do it. So the particles that are sticking together, hopefully they'd come apart in hexane. and you'd be able to measure them very, very readily.
without having to worry that water would interfere with the particle size. Okay, so back to the question about pharmaceutical industry. What about, can you comment more about tablet manufacturing in the pharmaceutical industry?
Well, I mean, that's a, we should have a whole talk on tableting, because, what I think the people in the pharma industry know very, very well is that getting the right tablet depends on, do you have a uniform mixture going into that tablet to start with, which is a whole nother problem in particle technologies. How do you maintain uniform mixtures? The second thing is in tableting, do you have the right compression? Do you have the right flow aids? Do you have the right particle size that actually will compress nicely? Uh, I suspect, I don't work in the pharma industry, but I listen to many, many talks on tableting, understanding every aspect of your powder, from size to flow to uniformity to moisture to the blend materials that you use, all are incredibly important and complicated. Keeping your material consistent from the start of your tableting process to the end, I think this is the challenge that pharma faces every single day. And for those of us that aren't in pharma, we face interesting challenges, but clearly pharma faces the toughest ones of all of us.
Especially with the FDA breathing down your neck. That's not easy. Okay. The next question is, and I'll honor your time. I'll take maybe 2 more questions and we'll wrap up. What is the best test method for nano-sized material in slurry?
We're typically going to use a dynamic light scattering system to measure particles. I mean, that's we found that consistently the best one to use. The one challenge about that is, do you have to dilute your slurry sample to get it to a low enough concentration to be used? And we all know that nanoparticles, when you dilute them, can change the aggregation. And so there's always that challenge. So, at the end of the day, if you're really concerned about, are you getting an accurate measure? I've heard of many people doing a freeze drying experiment and then doing an SEM image analysis to do a confirmation. But dynamic light scattering is the technique to use.
Thank you. And I believe that dynamic light scattering can handle quite concentrated samples, so there may be a way to go around it.
Mm-hmm.
I think that's about it. Is there any concluding comment that you would like to add, Willie?
No, this is a complicated area. Please bear in mind, you can't run enough particle size measurements, or you can't run enough characterization measurements. You'll go out and abuse your analytical departments. That's what I say.
Yes, for sure. And sometimes that means submitting samples to HORIBA Instruments, so we have a suite of different technologies here, or maybe at AVEKA, just to see,
Yep.
from different perspective, what exactly you're working with, what exactly the particle challenge is.
Sometimes that's what you need, another set of eyes.
So, perfect. All right, Willie, we look forward to your talk in 2023. And for those of you who are interested, please leave your information or sign up for our newsletter so we are allowed to send these information to you. For now, thank you so much for your time, and we will see you at our next webinar.
Thanks, Julie.
Thanks, Willie. Thanks, everyone. Bye for now.